Good morning, everyone. Welcome, students. We have already discussed bioprocess technology in our last two sessions. In today's session, we are going to proceed with some more information regarding bioprocess technology. Bioprocess technology is a fascinating field that involves using different living cells, such as bacteria, yeast, to produce valuable products. So, it has a huge application from pharmaceuticals, food production to environmental solution. Bioprocess technology plays a crucial role in creating sustainable and efficient progress. So get ready to explore the interactions of biology as well as technology in this exciting journey. So in our last two classes, we have discussed many things like what are the application of bioprocess technology applications and what are the small problem related to bioprocess technology, how we have divided bioprocess technology in three basic groups: upstream processing, bioreaction, and downstream processing. Those things we have discussed in a brief manner. Okay, let me share my screen for you. Just give me a moment. I hope the screen is visible to you right now. Okay, so we are talking about this bioprocess technology. The key aspects of bioprocess technology is to serve the nation with various possibilities. To serve the nation with various possibilities, the like key aspects are microbial fermentation, cell culture and technology, downstream processing, different genetic engineering, bioreactors, application in various industry are there. So we have discussed all of the points in a brief manner. So different example like production of insulin, biofuel production, those are the application of bioprocess technology. I hope you all have remembered that. Okay. So next, we are dealing about upstream processing. Regarding upstream processing, refers to the initial stages of bioprocess technology, where manufacturings are being taken places, preparatory phases are being involved. Those are called upstream processing. So initially, in our last class, we have discussed about media preparation. Regarding media preparation, yes, for the microbes to grow, many things are being required. As an example, we can talk about carbon source, which includes various carbohydrate, which includes oils and fats, which includes hydrocarbons. So that can be utilized for the resource of carbon in media formulation. Different nitrogen source can be utilized for that. Those include organic nitrogen as well as inorganic nitrogen for the production. Next, we can talk about different uh, um, minerals, buffers, precursors. Those are also being utilized for media formulation. One thing regarding media formulation was important. We'll come to that slide. Yes, this was the slide of media formulation. You can see here, media preparation is a crucial step in bioprocess technology for cell culture. It involves the careful formulation and sterilization of the nutrient-rich media to support the growth and viability of the cell. So, as I have said, media preparation plays a crucial role. You can see here 2-3 things are being written here. Careful formulation. That means careful formulation means you have to give your utmost care while choosing ingredients for the media. Next, just okay. So Next, we have talked about sterilization of nutrient-rich medium. So while you create the media, that's not that all done. Then you have to sterilize the media also. That is one important thing. And before we start with sterilization of the media, we have to, uh, I will go recapitulating these things like carbon components provide energy for the cells to growth, nitrogen source, they generally supply amino acid and nitrogen compound to the for the protein synthesis and cell growth. Then different vitamins are there, which serves as a cofactor for enzymatic reactions and plays a crucial role in cell metabolism. Then minerals and all, which provide essential ions for the cell and cells to create its structure as well as function, such as magnesium, calcium, these things. Okay, so media formulation is one of the crucial role. Then optimizing that media formulation, like we have to do optimization of that. I'll come that later. Before going to start that optimization part, we have to understand that on that media, what is going to work, like what are the microorganism which is going to utilize that nutrients. So the next important part in upstream processing is strain improvement or strain development. Strain improvement or strain development. That means a microbial strain is supposed to be chosen for certain purpose. Like Saccharomyces cerevisiae, I have chosen for the production of alcohol. Now I have to modulate the Saccharomyces cerevisiae in a manner so that I can get highest amount of alcohol at the end. So that will be modification. Another thing, another example will explain this better manner that initially penicillin was being produced by Penicillium notatum. Penicillium notatum was initially invented and penicillin was being isolated from there. Next, after few generation or after few years, there is a slight modification done in the genetic structure or the DNA structure in Penicillium notatum to create or to produce a high impactful penicillin, which can combat with various types of bacteria. So that modification done in the genetic structure of Penicillium notatum. Then the second generation, third generation antibiotics are being produced from that same microbes. But the microbes didn't remain same. Their genetic modification has been done, so their species has been changed. So nowadays we utilize, not exactly nowadays, before 20 years, Penicillium chrysogenum was being utilized as the source of um, penicillin G. Initially, penicillin was being produced. Now, next penicillin G is being produced. That was the second generation or third generation antibiotic. Now there are various modifications has been done in that penicillin production, and these are done in a genetic level. Okay, so genetic modification of strain can be done in various manner to improve the quality of that uh, microorganism. So what do you mean by strain improvement or strain development? What do you mean by that? Actually, the science and technology of manipulating and improving the microbial strain in order to enhance their metabolic capacities for biotechnological applications are referred as strain improvement or strain development. Okay, so we are supposed to modify or modulate the microorganism in order to get the best result. So that is known as strain improvement, and which is one of the most important part of uh, that upstream processing. Okay, so upstream processing include media formulation, it include strain improvement and many more. So we are going to discuss strain improvement in today's session. So I'm sharing my screen with you. You can see here these are the target of strain improvement. That means why I am going to choose strain improvement. What is the need? What is the need for strain improvement? The need for the strain improvement is that by doing so, we are trying to uh, capable the microorganism to grow rapidly. We are trying to grow the microorganism in a rapid manner. That means I'm going to improve the strain in a manner that it will grow very fast. As fast as it will grow, we will be going to get more product over there. Okay. The genetic modification should be done in a manner that the new gene structure should be stable. So genetic stability, one of the most important point that has to be taken care while we are going to modify the strains. Genetic stability. Then sometimes there are certain microorganisms which are very helpful and they can capable to produce certain product, and they're very good. But along with that product, they produce certain components which are toxic to human, That means suppose a microorganism is capable to produce Suppose the microorganism is capable to produce this product, and this product is very much important and good, but along with this product, this part is also been attached. This part is toxic to human, but this rest of these things are very important for human. So the microorganism generally produce two things together. They produce this part also along with this. So our aim is to modify the gene in a manner that the microorganism can only be capable to produce this which is important for human, and this the residual part or the other part which has been produced by the microorganism but not that much important for human, or we can say this is toxic to human, that will not get produced while will produce in a bulk quality. Okay, so these should be taken care. Large cell size for easy removal from the culture fluid. So we are trying to improve the strain in a manner that the cell size can increase, or we can choose a big microorganism or big or prokaryotes, except prokaryotes, we can choose certain eukaryotes, single cellular eukaryotes, but as because they are big in size, we can choose them and we can modify the gene structure for them so that they can be removed easily from the culture fluid, because as it will be big in size, it will be very easy to remove them from the mixed or mixture or that culture fluid. We are supposed to utilize or improve the strain because we know that strains are nothing but the microorganism. They require carbon source, nitrogen source. Yes, we are supposed to provide substrate for them, but sometimes microorganism required certain substrate which are costly. So our aim is to improve the strain in a manner so that the microorganism can utilize cheaper substrate. They can utilize or their ability to use cheaper substrate so that the production cost will decrease and the profit will be increased. That is one of the target to strain improvement. Next, we can talk about that elimination of the production of the compounds that may interfere with the downstream processing. We are going to choose this as an example that suppose this part is my product, this part is my product, but the microorganism while produce this, the microorganism is not going to produce only one component. They will produce many more biological components. So the microorganism produce these two, but the problem is when these two proteins are being produced, they create a complex structure. So what will be my aim? While we will be dealing with the improvement of the microorganism, we are going to try, or we will try to produce the microorganism in a manner so that they can produce only this. They can inhibit the growth of this. So elimination of the production component that may interfere with the downstream processing. That means at the end, while I am supposed to separate the end product, some components are there which are supposed to interfere, with that component. So our aim is to reduce or remove that interfering component. Okay. So elimination of the production, those components from that culture fluid is another important thing. Next, we can talk about increase the productivity. Yes, in every case, we try to manipulate the process so that we can get the increased product. Yes, in case of our studies, we try to create a good ambience for the student in class so that they can understand better. We try to provide various types of lecturers modules like certain videos, some photographs. We go for excursion so that a student can understand the subject in a fruitful manner. Similarly, while dealing with microorganism, we try to provide the best environment so that the end product, that means the productivity, will get increased. Okay. Next point, we can say that to improve the use of carbon and nitrogen resource, like investment is been done. Investment is been done in case of this bioprocess technology. We are supposed to invest our money to create media, to formulate the media. So that investment should not go in vain. That means microorganism should use the carbon source, nitrogen source, which has been provided to them in a proper manner, so that I must not face any loss over there. We must not face any loss over there. And whatever we have provided to the microorganism, they can utilize it highest part. Okay, that has to be taken care. Next, we can talk about reduction of cultivation cost, that lower price in nutrition. That means we are supposed to improve the microbes in a manner that the um, media components are very cheap and microbes can utilize that cheap nutrition source. We are supposed to modulate in a manner so that lower requirement for the oxygen, that microorganism can utilize the less oxygen which has been provided in the bioreactor. That should be sufficient. I am not supposed to buy oxygen and insert it into that bioreactor. That is again a tedious job. So that should be taken care. And during production of product, we are supposed to add certain additional enzymes, some certain addition mm, certain component which accelerate the growth of bioprocess, that generally increase the cost of production. So those are supposed to reduce. So we are supposed to modulate the strain in a manner so that that investment should be reduced. Okay. So these are the, we can say, target of strain improvement, like why we are going to do strain improvement. Now, how you are going to do strain improvement? Coming to the next slide, you can see here. These are the methodologies. These are the methodologies we can opt to improve the strain. So it can be done by optimizing the environmental condition. Yes, certain environmental condition, if we optimize that, we'll get a good end product. Like a microorganism require 24-degree for its growth, and I am providing 34-degree. It's not that okay, because the microbes will not grow properly and all my investment will be in vain. So I should try to keep the temperature in 24 so that microorganism can grow in a best amount. Similarly, we can talk about nutrient imbalance, like optimizing nutrition for the microorganism. That suppose a microorganism require good nitrogen source and I have provided ammonium nitrate as a nitrogen source. Now good. Now microorganisms are utilizing nitrogen in a very rapid manner and the nitrogen are being utilized. Now it is ammonium nitrate. So free ammonium salts are being there in a bioreactor and OH are there. So ammonium hydroxide is being produced and the pH of the medium are going to change, or it will, as we said, it will increase. So this kind of thing is called alkaline drift. The pH of the medium will be alkaline and this will be called as alkaline drift, and which can hinder the growth of microorganisms. So optimization in these area has to be taken care. Another example we can take that, suppose I have taken as a nitrogen source NH4 Cl, ammonium chloride. I have taken ammonium chloride and the microorganism is utilizing that that nitrogen in a very rapid manner, and free HCl are being prepared, it's created in the medium. So production of this HCl, what it will cause? It will change the pH of the medium and this will be marked as acid drift. This will be marked as acid drift. Due to this acid drift, what will happen? The pH of the medium will change, and the microorganism was familiar to grow in certain pH, and by chance, certain change or certain thrush in this pH change will create a uncomfortable situation for the microbes to grow, and their growth will be hindered. Their growth will be hindered. As their growth will be hindered, the production will hamper. So these are the correlated area. So we are supposed to take care regarding all these things. Now, microorganism and their strain can be improved by various methods. Now, some method include not involvement of foreign DNA. Some method include not involvement of foreign DNA, and some method includes the addition of foreign DNA into it. Those are the part of strain improvement. In which method we don't incorporate any foreign DNA, that is called mutagenesis. Mutagenesis is a method in which we are not going to utilize any foreign DNA, but the mutation will occur spontaneously, or we are going to introduce certain mutagen to do mutation. So that is a method in which we don't include any foreign DNA. Another method is where we include foreign DNA by means of various technology, genetic engineering, recombinant DNA technology, and other three important group of, we can say, recombination method is transduction, conjugation, and transformation. Okay. So I'm going to stop share the slide, and I'm going to share it once again. Yes. I hope this slide is visible to you. No. Slides are, yes. Now I hope that the slide are visible to you. Yes, thank you. Okay, so just give me a moment. Due to some network errors, it's taking longer than the usual time. Okay, okay. So everything is okay now.